Yes, okay. So good evening again. Apparently we have to wait about 10 minutes. There's someone who's stuck in traffic, a respondent for the presentation. Oh, okay, now is here. Okay, so we will soon be starting. Thank you. May we please rise for the academic procession. Please remain standing. The national anthem will be led by Ms. Kaylin Williams, soloist of the UWC Creative Arts Choir. Please be seated. I now call upon Professor David Holgate, Dean of the Faculty of Natural Sciences, to welcome you all. Okay, Prof. Holgate.
Thank you, Admire. I see we've intimidated you into taking your hat off. Welcome, everybody, from my side. So I'm here as Dean. I want to start with welcoming those who are not part of the UWC family. I think it's mostly this grouping over here who are here to support Tertius, not because you're a colleague, but because you have a family or personal connection to him, but especially to those people, a very warm welcome. To colleagues from the university, to Prof. Matete Madiba, our DVC, who's here, to the deans representing the other faculties, thank you for being here, and to colleagues and students. It's lovely to have you to share in this occasion. And of course, Tertius, welcome to you. We're looking forward to what you have to say just now. Reaching a professorship is sort of the pinnacle of your academic career. I see there are a few students around. I don't know how many of you are hoping to be professor one day. And it is certainly something that we aim for, and we're proud and pleased when we get there. And an inaugural lecture is really just a chance to recognize that achievement, but it's also an opportunity to share with others what it is that we profess, what it is that you've made your mark in. And so the pomp and ceremony is important because it acknowledges the central gravity, academic gravity of the occasion, and it's core to our academic project. So in that way, we put on our gowns and we show the respect. But of course, it's not an exercise in elitism or academic arrogance. It's really a chance to share what we are passionate about. So I hope that that's the experience that you have this evening, that you feel welcome, that Tertius will share his passion, and we'll share in it with him. And just to celebrate, to acknowledge together what you've achieved and the contribution that you make to the faculty. So from my side, a very warm welcome. I hope we enjoy the evening together. And we look forward just now, Tertius, to what you have to say. You're talking about elite athletes at some point, I think. I'm not sure how many of those are in the audience. But I'm sure we'll have something that we can identify with in what you say later. But thank you. Thanks, Advai. Oh, shall I call on Prof. Matete Madiba is going to welcome you. You may have looked at the program and thought she doesn't look like Prof. Monwabisi Khala. That's because she isn't. Unfortunately, Prof. Monwa has been in hospital, so that's a good excuse for not being here. And we're very grateful, Matete, that you're here. Thank you.
Tobela, good afternoon, or is it evening already? I can assure you Prof. Khala is recovering very well, so very soon he'll be back on campus. But for now, I have to stand in, and I wouldn't miss the opportunity. It is a real pleasure to welcome you to this lecture, because I've learned that when we gather in this way as a university, we are here, and I hear it's without arrogance, but we are here to showcase the very heartbeat of the university, the research that is happening in this environment, but also the kind of research that informs our classroom, because you cannot separate the two, research and teaching and learning at a university. So we are here to take stock of the depth of work that goes in, and we are very thankful that you have all gathered in this way and you have joined. And I just want to assure you that it's not just a moment of celebration for today. Of course, part of it is that celebration, but I learn it is going to be an intellectual workout. We are going to do some intellectual stretches and moves, but intellectual. And I can assure you, if you didn't stretch enough before you came, you are in the right place. And fortunately, the speaker is not going to charge you for any gym membership. But he's going to make sure that, you are intellectually stretched. Professor Kohn, we are so honored to share in this moment, to hear about your years of labor. Labor of love, but we know it is real hard labor to come through the ranks and end up being a full professor. We appreciate that, and you're going to be sharing with us and just trying to have a glimpse into your work. I feel like I'm one of those, I don't know about the rest of you in the audience, that after listening to this presentation, I'm going to regret it that I never studied biology. And you can see, so I might not have any chance to pivot. But those who still have a chance, students, you might want to consider pivoting into the study of biology and how the muscles work. Because somebody challenged us at another environment and said, because biology is about helping us to understand our bodies, and the speaker at that moment said, he doesn't understand why it's not one of those compulsory subjects, at school. But there are so many things that can be compulsory. I just hope that after listening to this labor of love that has been worked over time, over a number of years, you are really going to feel like, oh, I should have studied biology. So please come and help us to feel and to be exposed to the kind of work that you've been working on. Thank you.
Thank you, Professor Holgate and DVC Prof. Matete. I now call upon Professor Liana Marie, who is the Acting Deputy Head of Department of the Department of Medical Biosciences, to introduce Professor Kohn. Thank you.
Good afternoon, everyone. Thanks for joining us, myself representing our department, Medical Biosciences, here this afternoon. To all distinguished guests and the deans and the deputy deans and people standing in for others. We're very happy to have you here this afternoon. Also, all the staff and students from UWC, as well as friends and family. And also want to especially welcome Tertius's wife, Jolyn, and his two daughters, Jenna and Lara, here this afternoon. We're very happy to have you here and to join us in this celebration this afternoon. So I'm just going to give you a brief outline of Tertius's academic career and what it takes to be promoted to a professor. I promise I will keep it short. So in terms of Tertius's academic career, he actually used these skeletal muscles very well to roam around 3 different campuses here in the Western Cape. He made good use of Stellenbosch University, where he started as a BSc student and spent, I think about, my calculations might be wrong, but about 11 years going to postdoc status. Then he was moved on to UCT, where he was also a postdoc for 2 years, and then being appointed as senior research officer, which is similar to our senior lecturer, where he spent a couple of years while being at UCT. He also did an advanced postgraduate diploma in business management. Seems like he didn't have enough to keep himself busy. And then he was appointed here at UWC in 2019 as associate professor. And I must say, Tertius, I'm very glad you spent all the time at all the other universities and finally brought all the knowledge to UWC. And we're very proud as a department of this achievement of being promoted to professor. We welcome you as professor here at UWs. In terms of a promotion, those of you who don't know, it's quite a tough challenge to state your case for a promotion. You need 3 or 4 main categories, 4 main categories. You have to be excellent in learning and teaching, in research, community engagement, as well as leadership. So in all those categories, Tertius did excel, and I'm just going to highlight some of the things that he's achieved while he was not only here at UWC, but since his academic career. In terms of learning and teaching, here at UWC has redesigned and updated many modules that was outdated maybe for many years here at Medical Bioscience, especially focusing on exercise science for the third years, and also he has installed an elective for honors in exercise science that we did not have before. He's also very important for Tertius, I know, is the whole issue of academic integrity. So he has actually developed a plagiarism penalization scale, and he has engaged students to make use of AI obviously in a good way, not a bad way, to engage with different types of platforms. In terms of his research, he's a C2 rated NRF researcher. He has established the Mayo Lab, which is probably the only one in Africa. Now, those of you who don't know what Mayo Lab is, it's not a lab that produces mayonnaise for Spur or something. The Mayo Lab refers to, the Mayo prefix in biology refers to muscle. So it is a lab that specializes in research on muscles. He's had probably by now about 50 postgrad students. He has published more than 40 different academic papers on his research. So he is quite a well-established researcher in his own field. In terms of community engagement, he's got the Mayo Lab that's got a website. You're welcome to check it out. It's quite impressive. And it also helps to educate public awareness, or educate the public and cause awareness for his research topic. His research also featured in things like National Geographic, BBC Wild, and How Things Work, if you know of any of those platforms. And he's currently also involved in a study that monitors non-communicable diseases in a community in Santa Cruz. Then in terms of his leadership, institutional leadership, he's currently the chair of our Animal Ethics Research Committee, which is quite a task when you have a big department and a big faculty of science. He's also a faculty higher degrees committee member. He's our postgrad representative for our department, and we are a big department, so he's got a lot of work to do. And then he's also served on the local organizing committees of a number of international and national conferences. Tertius is also an extraordinary lecturer at the veterinary science at University of Pretoria. He has been an external course moderator and external reviewer for the NRF. He has also been an external examiner of many postgrad theses. So if you just look at that summary, I think he is well deserving of his position. So just to end off, before giving over to Tertius, I just have one request before you retire. Can you please, I'm an established long distance runner, but can you please make me run as fast as a cheetah? Right, thank you very much.
Thank you for that, Prof. Marie. I was hoping that you would call him up so that I wouldn't have to do this. But Prof. Tertius, I now call upon you to give your inaugural address. Thank you.
When you hit this age and professorship, this comes in handy. I'm just going to, there we go. Deputy Vice Chancellor and colleagues, deans of faculty, colleagues at UWC, Department of Medical Bioscience, students, ex-students, family and friends. So you're going to see a lot of that because I came prepared, and these things are tough. So welcome, and it is my privilege to present to you what I've been doing over 30 years. And sadly, none of my family could be here, my mom and dad and sister. So you're my family now. And welcome to my wife and 2 daughters. You'll be writing a test after the seminar. So And special welcome to Professor Tim Noakes and his wife, Marilyn. I really appreciate it that you are here today. What a daunting task of squeezing 30 years into an hour and 45 minutes. No, I'm kidding, it's 45 minutes. So, and as Liana said, you might actually know how to become faster after this lecture. And it's been an amazing journey, and this is what I want to tell the students. It's about the journey. It's not about the degree. It's about that journey, the friends that you make forever, those people that is like-minded than that what you are, and that shares your passion with you. And I'm going to show you photographs. Oh, some is nostalgic, but some of them you might recognize. And I see a lot of my ex-students also sitting here, and welcome to you all. So, as Liana said, my love and passion is skeletal muscle. It wasn't that way. I never thought I was going to stand here one day, and I don't call this a robe, I call it a cape. I always wanted to become an actor. Thank goodness I didn't. Apparently there's no money in it. But nevertheless, the journey made me fall into this field that I'm now. And as I mentioned, skeletal muscle was something that I didn't sort out. It became a passion the more that I learned about it. But I'm going to briefly just summarize what my field of research is, and then we'll delve into that nostalgia that really is such a, that brings joy to my heart. And when I teach the students, I also want that passion to say, wow, that's what you can achieve one day. So this is obviously one of the biggest crises that we have currently in South Africa and the world is obviously our favorite, obesity and type 2 diabetes. We are stretching around about 1 billion people in the world. And funny enough, some of our wild animals are also becoming insulin resistant. And I'll talk about that a little bit later. And these are all to do with exercise and muscle. The next topic that I do work on is exercise-induced pathophysiology. And that's basically when you're doing too much exercise and at too high intensity and you're not used to it, which leads to different conditions, but it also can lead to your death. Then, of course, muscle weakness. Muscle weakness is something that we are all going to one day have to go through. We're all getting older, and we're all going to get weaker. But specifically, when we look at things like when you end up in the ICU, the absolute devastation that that does to the body is very often irreversible. And we've done some work on that too, and I'll show you some pictures. And this is where my love for muscle actually came about, was looking at these absolute phenomenal athletes and then saying, but how do they work? How is it possible that someone that looks like a cricket can run so fast? How is it that someone so small can be so strong? And then I actually didn't pursue wild research. I fell into it, and you'll see how that came about. Many of you might have driven through it and didn't realize, but I was born in 1975 in a little dorpie called the Dorens, and this is the Hicks River Valley where all and most table grapes come from. And I was part of a family. And what you don't see, this little one here, this little guy, he's wearing gumboots and it's red. Now you can imagine what it's going to look like in this cape of mine. But I love, and from a very young age, I love taking things apart. This is my family. This is where my name comes from, by the way. Tertius means third. And my grandfather came from Poland, who lost his whole family during World War II. And I was the third Abraham down the line. But we're trying to fix a broken lawnmower there, and we took it apart, and that just fascinated me. And since then, I started taking things apart to find out how does it work. And that's where the biology comes in with regards to muscle. I started off my education in the Dorens, and then, yep, that's what happened when you get to matric. You become very arrogant. You know everything. and ended up with a PhD in skeletal muscle, in biochemistry of all things. In those days, you couldn't jump between departments. You actually had to get the degree of what you eventually studied. So I got a PhD in biochemistry in the physiology department. I am actually a musician as well. So that's me starting to learn how to play the guitar. But from a very young age, I actually started composing music, and I still do till today. So rock on. And I am absolutely infatuated with Superman. Not the new Superman, the good old Christopher Reeve Superman, who could actually make you believe that you can fly, because Christopher Reeve was a hang glider. He was a sportsman, so he knew exactly how to keep his body in an aerodynamic point. So hence the suit. So I always wanted to fly. And I was telling a colleague just now that in the Durans we only had rugby and rugby and rugby, no cricket, no tennis, a little bit of athletics here and there. So I then ended up going to Worcester, and I ended up doing gymnastics. So jumping on that trampoline, flying was amazing. So that was the closest that I could get to Superman. Tim, I did try running. This is me doing a half marathon during the SAD with no training, and I couldn't walk for 3 days. And that's when I decided, no way, running is not for me. So how did my journey at tertiary education start off? Well, my dad said to me, please, would you consider doing medicine? Because he did, but he passed out in his second year when he saw the dead body. So he wanted me to do it because I didn't pass out. But I wasn't a very good student. Students, do not listen now. I wasn't a very good student. So we started off with a BSc gewoon, and I ended up majoring in biochemistry and physiology, eventually doing honors in physiology on a very weird topic, Angora goats. And that was the trigger where we had to go to the Maitland Abattoir and get some tissue and blood and all that, and that really fascinated me. And one of my honors classmates are sitting right over there today. Hi. And then biochemistry didn't have a project for me, and then I remember this little tiny that was in the physiology department. She was about yay big, and then she said, why don't you come and do a project with me? And I didn't know that Kathy Meiberg was actually South Africa's best Springbok gymnast at that time. So she then enrolled me, and I was very excited. And she said something, you're going to do some muscle and sporting performance, and what did I know about sport and muscle adaptation. But the thing that she did say, you're going to have to go overseas, and that's all I heard. But did I remember anything about muscles and exercise? Zippo, nothing. So I had to learn everything from scratch. And that was my journey from 1998 to 2005 when I got my PhD. But what was the PhD about? And we basically made up the PhD as we went along, because that's how science works. You go for a hypothesis, and then suddenly you realize, oops, we're at a dead end. We need to turn around. And probably this photograph I use quite a lot of all these great athletes, but it has become a multi-trillion-dollar industry. I mean, we just have heard of another athlete that's been suspended because of doping, and that's just the challenge and things out there. Now, all these sports are amazing, and who doesn't like to watch gymnastics on television or Usain Bolt running that 100 meters? And then a study came out in 2000 by Bengt Saltine and his co-workers about muscles and genes. And that's not the genes I'm wearing, it's the genes that you and I harbor that we get from our parents. And the question was, but how and where? How do we identify and talent ID these athletes? Very often, it is still very difficult today. So this is the current world champions when it comes to holding the records. except sadly the current world record holder for the marathon sadly passed away, but the Kipchoge did break the 2-hour marathon. But what I want to show you is that if you look at the speeds these athletes are running at, that's phenomenal. If you don't know what it is, just go to a gym and put that treadmill to about 15 kilometers an hour. Then we'll talk. So Kipchoge can do that for 2 hours straight, running at 21 kilometers an hour. That is phenomenal. So the muscle and how they can actually sustain that endurance is just amazing without it collapsing. So what determines our performance capacity? And that is our intrinsic factors, which are primarily our genetics, like I mentioned, our parents, physiological, biochemical, anatomical, and of course psychological. I put there the WWW, and that's what I heard in school, is the will to want to win. Because if you don't believe you're going to win, forget it. Then you've given up already. And of course, as we know, with science these days, we do discover more and more things, and that's why I put there other. Then there's external factors, like exposure to a sport. You know, I told you, the Durans, rugby, but I was exposed to gymnastics, and then suddenly I excelled. I was not the bull anymore. Training, the amount of training that you require is going to have an effect. Environment, we know altitude, nutrition, socioeconomic status, technology, AI, I question mark that because maybe you're going to ask ChatGPT, please run the marathon for me, you never know. I know there's a robot Olympics currently somewhere, which is quite amazing. And then, of course, doping, and then other. Now, this picture I also like is that, you know, that we're not all made for the same sport. So that's where those intrinsic factors come in. So I can imagine that, you know, if you look at these statures, each sport has got their own where you have an advantage to a sport. For example, I will never be a great basketball player, never, even if I can fly. And the woman isn't far behind. And definitely those things will identify what sport you will have an advantage to. Usain Bolt, for example, was actually very tall compared to all the other athletes. So how do we test human fitness? Well, basic in a nutshell, we put them in a treadmill, we stick a mask on his face, and then we measure his oxygen consumption and carbon dioxide that he exhales, and heart rate. And from that, we can actually determine how much oxygen they use, what's their maximum capacity that they can use, and what fuels they are burning. Are they burning fat? Are they burning carbs? Are they producing lactate, etc.? We can do the same on a single muscle fiber. And this is where what our lab makes so unique in Africa is that we can do all those tests that we do on a human, we can do on a single muscle fiber. To give you an idea how thick that fiber is, if you take your hair and divide it by 2, that's one of the thickest fibers that we will get. And that little thing can make you run like the wind. So for that, we obviously need to take a muscle biopsy. In humans, we stick a needle in your leg and then we take it out. Or if we do work with animals, we do an open muscle biopsy, because then there's less trauma to the muscle. So now I want to quickly recap, kids, this is what the test will be about, is how does skeletal muscle work? So this is quite phenomenal. So by the way, those black lines that you see there, those are your sarcomere spacings. So those lines is the ones between 2 lines, that's what make you able to walk, to write, to run, almost said fly, but not yet. So how does it work? The sarcomere, as I mentioned, those black lines, the blue ones, they're the ones who would be able to, almost like an accordion, pull towards one another when you have a fuel source. And how that happens is your brain tells the muscle you need to contract, so there's a nerve impulse, and that releases calcium in the muscle. And for that you need ATP, which is our energy source, which comes from the metabolism. And eventually that little muscle fiber is going to contract, or that sarcomere. But it is like microseconds. That's how quickly it can happen. And then, of course, very important is that if you want to relax your muscles, because you cannot be in a spasm the whole time, the calcium is taken back up into the sarcoplasmic reticulum, and for that you also require ATP. So there's a lot of energy that needs to go into the muscle to contract, but also to relax. If you do not have ATP, you get a condition called rigor mortis. So, this is for my Formula One friends in the audience. So, I love comparing it to a car engine. I'm not advertising any brand. So, on the left, you'll see the muscle fibers, and they hone down into smaller parts. We have the myofibrils, and eventually those yellow and red parts, that's the myosin and the actin that actually makes the muscle fiber contract. Those little pink things, they're the mitochondria, very important, because they're the ones that can generate ATP very efficiently, and I'll show you some of that a little bit later on. So like a car engine, we all know, this is an internal combustion engine, not these new hybrid electrical motor lithium ion things. Kind of maybe the same principle, but this one will do. We all know that the bigger the engine, the more power and the more force it will generate. So with muscle, it's the same. Bigger the fiber, the more force it will generate. But also the fiber type. The type of fiber that muscle harbor is also going to have an effect, and I'll talk about that a little bit later. Then, of course, the fuel. If you've got an engine, but there's no fuel, it's not going to run. And that very often we see with congenital myopathies where a certain fuel cannot be utilized. Combine it with with oxygen, and those 4 fuels, phosphocreatine, glucose, glycogen, and fat, and it can supply the engine with ATP. Then, of course, the exhaust, very important in muscle, getting rid of the carbon dioxide and lactate. Otherwise, it can have a negative impact on the engine itself. And then what do we measure in single muscle fibers? That is, we measure force and how quickly that fiber can contract. And when we combine the two, force times velocity, we get power, because power is the one that we're really interested in. And then, of course, economics. I always tell the third-year students that if you want to be economical, you drive your car at 110 kilometers an hour, much more fuel efficient. So those parts that we talk about now, the fuel, the metabolism, the ATP demand, and the ATP supply and the exhaust, that's what this talk is going to focus on. One day my student came to me, I was still at UCT, and they said to me, "Look at this." And I said to her, "I've never seen this." This was actually ostrich muscle. And basically, she said, "Look how red these fibers are and the other fibers are white." And I said, "Wow, that is amazing. Why is that?" And the reason for that is because some of the muscle fibers store more myoglobin than the other fibers. And the myoglobin is important to show or to store oxygen. So that red fiber, that's where the original fibers got their name, red oxidative fibers and the so-called white glycolytic fibers. And that was just amazing. Took it by its cell phone. I think it was a Nokia 3310 or something. These days, we use antibodies to specifically stain for the fiber types. And the blue ones you'll see throughout this presentation is the type 1 fibers. They're the slow twitch fibers, usually what our endurance athletes have. The green ones are the 2A fibers. They both fast and they have a lot of endurance capacity, whereas the 2X fibers, they're the red ones. They give you a lot of force, but they fatigue very quickly. I'm not going to spend too much on this because I can already see it's becoming late in the evening, but basically we use certain markers to give us an idea of the capacity of a specific pathway. So in our bodies, we have, as I mentioned, we have 4 fuels that we use, and each of them slot into a specific pathway. Think of it this way, your petrol tank and all those pipes and things that it goes through. That's the metabolism that we're talking about. So the bigger the pipe, the more fuel can go through. And the enzymes that we use, they can give us an idea of the flux, how big is that pipe. So these enzymes, you're going to see them later on in the talk, but I want to highlight why they're important. So first of all, Phosphocreatine is probably one of the most inefficient fuels around. For one phosphocreatine, you only get 1 ATP. And as we go down the line, you'll see that fat is actually a very efficient fuel. From one palmitic acid, you can generate 120 ATPs when it goes through the Krebs cycle. And that is something that our athletes really would require during running. We can stain for the mitochondria by using a simple NADH stain. So we take a histology and we then stain it, and you'll see that each of these fiber types have got different mitochondrial content. And that is also important, especially when we talk about the wild animals. Kids, you're going to write a test about this table? I'm kidding. So our endurance athletes, they primarily have these slow oxidative type 1 and type 2A fibers. And our sprinters usually have the 2A and 2X fibers, a predominance of that, but they also have type 1 fibers. So I'm just going to talk about the type 1, 2A, and 2X fibers going forward. And then a study came out by the Copenhagen group in Denmark where they investigated the difference between Kenyan runners and Scandinavian runners. Now, to not go into too much detail, the thing that stood out about this study for me was the extraordinary lactate dehydrogenase activity that these athletes had. Now, even when some of the Scandinavian runners went to altitude to train there, their lactate dehydrogenase decreased. And you'll see now why that is so important, especially when it comes to the wild animals. So Kathy Meiberg said, off you go, you're going to Denmark, land of the cheese, apparently, and The Little Mermaid. And I worked with Bengt Saltine and Jesper Andersen. And they taught me how to do these single fiber dissections. It's also the home of Lego, but it's too expensive there. So there you have the first single fibers that were dissected in South Africa, freeze-dried single fibers, and if you can't see them, there they are. They're like little freeze-dried 2-minute noodles, and those you can handle very well, and you can then do experiments on them. I think this is where the nostalgia came in, and this is my colleagues from when we were studying, is the laboratory that we had at Stellenbosch. This is the one on the left, of your left, and then they revamped that building, and then we were temporarily housed in the food science, very important, food science building, and then we moved into our new building, which I'll just show. And then if you look on the bottom right slide, you'll see the dissection kit, as well as a 7-decimal balance. So you could weigh off very, very small quantities of chemicals, but also the fibers, because you had to weigh off in micrograms. These were the new laboratories that we moved into in 2002, and there is the small exercise group that we had during that time. Then Kathy said, off you go to Uppsala, Sweden, because we now have the single fibers, but we now need to know how to measure the metabolites and the enzymes in those various fiber types. And that's the first time I saw a horse on a treadmill. It is really something spectacular to see while that horse is running. And some of my colleagues or students here that has undergone a muscle biopsy now, that horse just had a muscle biopsy. That's that white plaster you see on its bum. And it was doing absolutely fine. And that's where my prickle came for animal research. That lady that you see in the picture, and I hope she's watching on the YouTube video, is Birgitta Essen Gustafsson. She was my Swedish mother because she looked after me very, very well while I was there. And then we have all those little fibers over there. It's the first time I ever set foot in snow. And believe you me, it's freaking cold. Minus 25 degrees Celsius, hence the picture on the bottom right. My hands started freezing, and I had to run back inside. But it was fun. And then Kathy also came to visit. So what did we do? So first of all, we compared runners with untrained runners, because when you do science, you have to show that your model is working. Then your methods might be wrong. So we decided, okay, we're going to have untrained individuals, and we're going to compare them with trained runners. But not your, I do a bit of jogging. We wanted pretty good runners. Let's call them sub-elite. Abraham, you can do that time, right? 33 for 10K. Yes. But these guys were spoegspot, so they were really very good. And we compared them with one another. And as I mentioned, we slapped a mask on them, put them on a treadmill, and we made them run. We started at a very low speed, and then where they cannot run anymore, that will be their peak treadmill speed. And as you can see, there's a big difference between untrained and trained runners. High VO2 max, high peak treadmill speed that they can achieve. If you didn't get that difference, then your athletes weren't trained, by the way, or they were lying, which we actually did get. Now, if we look at the metabolism of our athletes and untrained individuals, especially the CS and the HAD, now CS is a very good marker of mitochondrial volume and content in our muscle, and with training, endurance training, that enzyme will increase. So you will actually get more mitochondria and more of that enzyme, as well as the HAD. And that HAD is for beta oxidation. And the beta oxidation is there to produce more and more ATP. And then we found a significant difference, which was great. Oh, by the way, my emojis aren't working very well, and I'm not sure. Sometimes there might be little blocks. But nevertheless, then we looked at the glycolytic capacity, because in conventional theory is that those glycolytic and lactate dehydrogenase activity will actually decrease with endurance training. And we saw that it didn't. And that made us very worried. What is going on? The same with the fiber type. As I mentioned, that if you're an endurance runner, you gain more type 1 fibers over the years. But there was no difference between untrained and our trained runners, which is going on, what is this? We then had to really think very hard, and we thought, okay, what if it's genetic? You know, what if there's some individuals that just more prone to have a higher type 2A fiber type or higher glycolytic capacity? Or can it be something else? Could it be training related? And we realized that actually when we recruited our athletes, we recruited them from 2 different clubs. And as you know, me being gymnastics coach all my life, well, up to 30, and I couldn't anymore, basically that a coach has his own methods and will bestow training on all their athletes all at once. So whether you're doing fartlegs, or whether you're running in the sand, which is high intensity, or just doing long runs. So we decided, okay, let's break them up. But it happened to be that they came from 2 clubs where there was a demographic difference. And there was this clustering of data points, which is very important. That's one thing Kathy Mybeck taught me. You must look at your data points. So we then divided them up into African Runners Club and European Runners Club. And we found that the athletes that were from the African Runners Club had extremely high lactate dehydrogenase activity, and again, made no sense. Whereas the so-called European runners, they conformed to the conventional studies that were what we found has been published. Now, you think of the Kenyan and the Scandinavian runners' details. Exactly what we also saw was that the European runners have more type 1 fibers, whereas the African runners had more type 2A fibers. So, quickly, and then we found a very strange phenomenon as well, and that's that little emoji with a question mark, is that usually when you have a lower or higher activity, you should be able to produce more of a specific product. Not in this case. The plasma lactate during exercise actually decreased, which made no sense. And when we looked at single fiber enzyme activities typed according to their type 1 or type 2A fibers, our friends from the African Runners Club had actually much higher activity. So this caused a bit of a conundrum. So that's why I put this question mark. So why? What can be the reason for that? It makes no sense. And this might have been indirect evidence of the so-called proposed lactate shuttle hypothesis that the third-year students will be doing in my course. But we didn't get that evidence. Or could the type of training merely have been different, where our African brothers basically just trained harder because of the coach? So my athletes said, more muscle biopsies, bring it on. Let's get some needles into us. So what we decided then was we're going to train all these athletes ourselves, bring them under one roof, make all the playing fields level, and then we're going to give them super duper spook spot training on a treadmill, because then you can control the speed. And by the way, it worked very well. So just to give you an idea, remember I mentioned 20 kilometers an hour? So we had, for example, our athletes on average ran at 20 kilometers an hour for about 3 minutes, and then they only had 1.5 minute rest. And they had to repeat that 6 times, and they came in twice a week for only 4 weeks. So what did we find? And remember, these are well-trained athletes. Apologies for the poor resolution, but in 2003, there wasn't very good camera quality. This is one of the athletes that were running on the treadmill. Currently, it's running there at 21 kilometers an hour, and found it quite easy because he was already well familiar with it. And then just wanted to show a photograph of all the athletes that took part in that study. And Tim, do you recognize one of the athletes there? Oh, I don't have a pointer. It's Johan Landsman. Yes, so Johan Landsman standing there. He was also a phenomenal athlete. Right, so what did we find? Well, 4 weeks of training, and their peak treadmill speed increased with 1 kilometer an hour. That is quite significant for only 4 weeks of training. The VO2 max didn't really change, but what we did see was the following. And that is that their heart rate decreased at the same running intensity. The economy improved. This is by only basically putting in 10 minutes of extra high-intensity exercise, and the plasma lactate decreased at the same intensity. What happened to the muscle fibers? Well, if you look at the middle graph, you'll see that the type 1 fibers kind of tended to decrease, and when we looked at the other fibers, the 2As, it wasn't really different. But the hybrids, the 2A, 2X fibers started increasing. And the hybrids are fibers that are, we believe it's transitioning from becoming a 2A to a 2X. So they're actually becoming faster twitch fibers. When we plotted the change between type 1 fibers and the speed, we showed that as the speed increased, where you trained at, you actually started losing type 1 fibers, which is a good thing. And when you do plot it against the 2AX fibers, you see a beautiful positive correlation. Now keep that in mind, please, because when we get to the wild animals, you're going to see something quite phenomenal. But then here again, they can produce more power, but all the fibers shrank. So you now have smaller engines, but yet they can produce more force. And that we still don't really understand. We then looked at this lactate dehydrogenase enzyme, and we dissected a number of fibers out of it, and then we measured that enzyme activity again, and we found that after the training, it actually increased. It also increased in a fiber-specific or fiber type-specific manner, and when we plotted it against the actual speed, we also saw a positive relationship. So the answer is, if you want to be fast, you need to increase your lactate dehydrogenase activity. So the take-home message from that study was different running clubs implemented different training methods. I mean, I was a coach, I implemented, all my 8-year-old gymnasts had to do 100 push-ups, once off, no stopping. And that's just how different coaches approach that. So it was very well that the intensity of the exercise differed. So to become fast, you need to train at a very high intensity. You need more type 2A fibers. It makes sense. But the higher lactate dehydrogenase activity currently doesn't really make any sense. And what I want to show you, this paper came out 3 weeks ago where they basically gave guidelines of how to become fast if you're an endurance runner. And they are advocating exactly the exercise intensity and the ratio that we have done in 2003. That's the PhD. Whoops. That's the PhD. Got it. Yay. And then I wanted to go to America, live with my sister, but, get the American, maybe become an actor. And then she walked into my life, and she's been a wonderful wife to me, and we got married within 2 years. Yeah. They say when you know, you know. Eh? You know. I waited long enough. And then this is my gymnastics club that sustained me through my studies in the Strand. I could at least do an LSAT, the one on the bottom right. And I've been to some of their weddings. And again, my journey it's about relationships with people. While I was doing my PhD, I also got involved with also falling into it. I call it conversations in a parking lot. And where someone said, that stuff you do on humans, can you do it on animals? I said, well, I can't see why not. Yeah, we've got this wild animals. Let's do some research on them. So we did. And then we also did the first ostrich study where we put pedometers on their legs to see how much exercise they do. Luckily, we got it for free from Discovery. And so we did some research on those wild animals, mostly for the meat and the export industry. And then I had to leave Stellenbosch University. And very often, it's very sad to leave. You don't want to go. But God takes you into a specific route. And I had to go to UCT. Didn't want to, because the temperature is colder there in Stellenbosch, but I had to. And that's where I met a wonderful man by the name of Professor Edward Ojuka. And he's been an amazing inspiration in my life, the 2 years that we were together. And he introduced me to metabolic diseases, obesity, insulin resistance, type 2 diabetes, and cellular signaling. And Ed was a very quiet-spoken person, and when he spoke, everybody listened. So we did a couple of studies together, went to America together. It was great fun. And Ed actually studied under the great John Halsey, who also was the founder of mitochondria and exercise performance. After 2008, I started a new adventure with Professor Tim Noakes. You call me now? All right. So Tim was amazing. You can walk into his office and he's like, come, sit. And I said, what do I do now? My postdoc is finished. He said, no, you're going to stay here. I said, all right, that's great. Can you pay me? We'll make a plan. So I said, okay, I'm going in faith here. And Tim said to me, you must study wild animals to better understand human muscles, because we don't know a lot. And you must study weakness. And that's why I got involved with Red Cross Children's Hospital. And then eventually got into Tygerberg Hospital, where I met Dr. Frans Lehenen. And all those journeys. And then find the diseases that wild animals get that links to human health. Now that was a daunting task. You don't think that wildlife can actually get diseases. But lo and behold, our friends in the peninsula are currently eating themselves into type 2 diabetes. So we did a bunch of research on these animals, on the baboons that are just consuming, they fight, they will go crazy for our human carbohydrate-ladled foods, and we showed that they are becoming insulin resistant, and we need to do something about it. During my time at UCT, I was also very fortunate to work on the low-carb, high-fat diet and show why the low-carb, high-fat diet athletes appears to be insulin resistant. but it was actually because they're not used to taking in sugar, and that we published in Journal of Physiology and so forth. And then this was probably the key moment, that eureka moment where you see the link between wild animals and a disease in humans that we don't understand. So the athlete on the left is called Corey Stringer. He was an NFL rugby player, and he died of a condition called exertional heat stroke. And that condition wasn't recognized at the time. And his wife sued the NFL for a lot of money and won. So because it's such a disease that you can't really recognize, studying it is very difficult. And then Tim said, find a disease, that is in the back of my mind. And then I stumbled on this condition called capture myopathy. It was actually Eric Harley, who I chatted to, who just put this word out there, and I had no idea what it even means. Remember, I come from the Plus. English was a foreign language to me. I had to learn it overseas. And then, obviously, our president also is a wildlife farmer, so he's not going to be happy if he loses a very expensive buffalo, because sadly, once these animals present with that disease, you have to very often euthanize them. But the symptoms between these two, the exertional heat stroke that humans get and capture myopathy, is to the T the same. And from that, we did some very cool research. The student who trained Blaise Buck, and I'll show a video of that sitting at behind there. And I think this is the sadness that I could never have met Margaretha Hofmeyer. Many of you might know her. She was at BCB here in the faculty, and she sadly passed away just as I arrived at UWC. And she was a pioneer in this research right in the 1970s and 80s with Peter Bologna, who was also quite renowned scientist. So I thought I just wanted to bring her in honor of her work that she did. So one of the key treatments that can be used in wild animals in humans when they do get exertional heat stroke, and if you identify it, is by cooling them down. So they submerge you into ice water and wait for your body temperature to go back to normal. It's very difficult in wild animals because usually they're running around, they want to get away from you, kind of thing. It's a fear factor. And we tested that hypothesis, whether cooling will actually have a difference or make an effect in our wild animals. And I have to brag, my daughter drew all those Blaise Buck within almost 5 minutes. So we have a creative artist in the house. I can draw a straight stripe, that's it. So, but what we found was actually the cooling had a devastating effect on the muscle glycogen repletion after the exercise. And it's now something that we're pursuing further, because that could actually mean that these animals are glycogen depleted, and that's what's causing the severe muscle breakdown. This is a study that Dorothy Breed took on while 32 weeks pregnant, machine, and she trained 20 Blaise Buck up in Nelspruit, and then we chased them around to see whether to induce hypothermia. And that data we're going to publish very soon, but what I want to show you is the phenomenal team that took part in that study. All of them veterinary experts from all over the walks of life, one from Estonia, a couple from America, and all that. And we came together and we were able to show and train and exert those animals. When we studied muscle weakness, we had one patient that had inclusion body myositis, and he basically gave up his body and said, do whatever you like with me, stick things into me, which is always nice to hear from a participant. I hope he's listening, because I did send him the link. And we showed that when he does do some exercise training, that he does become a little bit better. Unfortunately, with his advanced age, it does tend to be more difficult. Then I was able to also work with Alejandro Lucia, who also was an exercise scientist and a medical doctor, and specializes in McArdle's disease. And we were able to do some really cool work with one another. And then Professor Franz Lehenning, who was the neurologist at the time at Tygerberg, and I was going to chat to him, let's collaborate or whatever. And then he said, can I be your student? And I was like, oh, I didn't expect that. Okay, why not? So he got his PhD in contractility in contractility in inflammatory myopathy. So he dissected out a lot of single fibers. But what about exercise performance in humans? Because that's what this talk is about. So if we look at this graph, and we've plotted the records over the years from 1890 right through to current, and we want to go to, and those are the current world record holders of the 100 meters, we see that there's this gradual, for the mathematicians, exponential decay. And when are we going to, and how, this when and how, legally and legitimately break those world records? And that is where the wild animal performance comes in. No one can tell me that this is not one of the most beautiful animals out there. And if you look at that speed that animal can run, is just phenomenal. And what you did hopefully notice is that the prey can also run that fast. So who's faster than a human? Well, basically everything. You know, you try to catch a cockroach, it always gets away, you know. So if we look at that, it's worthwhile studying these animals. And believe you me, there's so little out there, and we don't know even the little bit about them. So I did some research on how many papers are out there and animals that have been studied, and this includes humans, about 20 mammal species, and 12, and I call that complete analysis, where you look at the performance of the muscle, the metabolism, as well as the structure of the muscle, only 12. And we're sitting, and I saw, this is fantastic. We live in South Africa. We've got the big five. We have one of the best veterinary hospitals around in the country. Let's start getting some work done. And over this year, since 2000, and actually that's 2005, we've been able to do the muscle composition of about 15 different species. We've done pangolins, we've done baboons, we've done cheetahs, lions, caracals, etc. And it was just, each species is different, which is unbelievable, and we think that a cat is the same thing. So there was we got a couple of publications. So what I want to show you quickly, and if you're falling asleep, that's okay. But what I want to show you is the difference in the muscle when you just look at it. And on the left, you have an animal. It's an antelope, and it's got very dark red muscle. So those who love biltong, that's where your biltong is coming from. It's blesbok, by the way. And on your right, you have a lion. Now, you can see the difference in color of those fibers. The one on the right is very white colored, and then the one on the left is red, dark red color. So the one automatically will assume has a lot of myoglobin, endurance capacity, because the one in the middle is an endurance athlete muscle, red, and it has endurance capacity. And automatically will assume that the red muscle will have a lot of type 1 and 2A fibers, which will go a little bit against what these animals are capable of doing. And then on the other side, we have the lion that will be predominantly 2X. So this is some pie charts that we drew up to show you the difference, and we compare it always to humans. It is so important to compare it. We have a distance runner, and then we have a sprinter, and you'll see that the distance runner has very little 2X fibers, a lot of type 1 and type 2A fibers, whereas the sprinter, a little bit of 2X fibers. Anybody seen a sloth move before? What's that movie? Zootopia, am I correct? Zootopia, yes. So just, if you don't, you can go and watch it. Predominantly type 1 fibers, and the pangolin, funny enough, also has only predominantly type 1 fibers. Baboon, very similar to us, our fiber types. Dog, slightly more type 2X fibers, hence why they can run a little faster. Horse, also kind of 33%. And then if we start looking at these the cats, you'll see as they become a little bit smaller, they actually have more and more type 2X fibers. But funny enough, the antelopes, we thought they were going to be predominantly type 1 because they are so red in color. They actually very often have more type 2X than the lions, and that made no sense. The pictures on the left are histology sections. The one on the top is a South African top judo athlete, and the fibers on the bottom is that of a cheetah, pure red basically. So we then go, wait a minute, what's going on in these antelopes? This is very weird. So again, I'm bringing up the slide that shows you mitochondrial content in these in humans and as well as the animals. That is an endurance-trained athlete, very dark fibers, which represents a lot of mitochondria, and that's what you want for the endurance capacity. And we started staining the antelope. We saw, oh my word, they actually have a lot of mitochondria in there, but yet they've got a lot of 2X fibers. And if we bring the lion and the caracal, we see they have very few mitochondria. And that maybe is a good thing for the prey, because the lion with endurance is not something you want to mess with. So at least the antelope has some chance. They just have to run fast enough and last long enough. It also explains why lions and cheetahs and our cat species has to first stalk their prey, because they're going to run out of fuel within 45 to 50 seconds. But we made another discovery, which was quite cool, and it is a fourth fiber type, actually a fifth kind of one. I'm not going to talk about the fourth, but it's the so-called 2X oxidative muscle fiber. We haven't seen that before, and the antelope has that. So that means it's a fiber type that is extremely fast, that can produce a lot of power, but it has endurance, and the lions don't have that. So when I published this paper, I got phone call from some of the East Bloc countries. How do I get the 2X oxidative? And guess who the coach was? It was a boxing coach, because a boxer needs strength and endurance. So amazingly, I said, I don't know. I don't know how you'll do that. I think this graph kind of summarizes what we found so far. And Liana, hopefully this will answer your question. To be fast, you need a lot of 2X fibers. And that's a question that we are asking. Why are humans not able to just like 5% more 2X fibers, and you're going to run like the wind? But maybe the answer lies there, is that our training is not quite optimal. Because remember, when we trained our athletes on the treadmill, first they couldn't finish it. I mean, they were passed out. But after a while, they got used to it. Their fibers started changing. And maybe that's something that we need to focus on. This was also a surprise find to me. And basically, it's showing that if you want to be fast, you need a very high lactate dehydrogenase activity. And the question is, to what capacity can we push our athletes to have that activity? And yet, we see that when we train our athletes with a high-intensity interval training, that activity does change. But there's a threshold. So if you do too much low intensity, you're going to switch back to type 1. I think it's appropriate to just show you this picture. This is, basically I built most of the stuff myself, but because I passed woodwork, so, But the point is, this is the only machine in Africa where we can manipulate a muscle fiber. We can give it drugs to see, does it do this, does it do that, on a single muscle fiber level. And this is what we see there on the bottom left. That is one human single fiber, and you can see the striations there. It wasn't my fibers. And it's to see what can that fiber produce. And we then did the first single fiber study on lion muscle. And in a nutshell, if you look on the left, there's a comparison between human fibers and a caracal and a lion. You'll see that it's very, very thin fibers, but yet that thin fiber can produce 3 times more power than a human fiber. And the question is, why? So the take-home message is that we've made some very significant discoveries in the wild animal kingdom and more. We're going to have to do some more of that. But the similarities that we're finding and the questions that now arise, and this is probably where things like doping might come into play, is can humans increase their lactate dehydrogenase activity? Because we're seeing this beautiful correlation. And can humans, to some point, increase their type 2X content? Why can't we? So I'm going to wrap up now, but I want to just quickly, briefly mention my journey from UCT to UWC. And when I first stepped my feet here, I was like, what is this place? This is amazing. I have never seen laboratories like this. This is amazing. And I thought, wow. And if you look at that photo of the campus, it is just such a beautiful campus. So I arrived here in September 2019. I think it was about March 2020. They said, go home. And COVID hit. And we were in trouble because one of my other PhD students, Catherine von Boom, had to finish her PhD. So we decided we're going to go to Oudtshoorn and we're going to study cheetah nutrition because they also, we don't know much about their nutritional needs. They don't only need just meat. They also need other nutrition. And then on the left there is Lukman Adamson, who received his MSc summa cum laude on that same study. And we've managed to publish a few papers. While we were here, we also did some ICU studies at Tygerberg during COVID. It was quite daunting doing it with masks on, and that was a team, and I'm very grateful that I was able to work with such an amazing team of people. We're currently doing carbohydrate study where we want to revisit all these things that we found over the 30 years that I've worked with. I'm not going to go through all of that. All those little needles or syringes that you see, that's actually finger pricks. And then we are very fortunate to have state-of-the-art, brand new equipment that we can do our testing on. And in the future, we're going to also start looking at energy expenditure. Because it is something that what we want to do is to actually predict when is someone at risk. And very often we catch that diabetes, the insulin resistance, we catch it too late. Then the job's done. So we're focusing on young students. And I must say, from what I've heard from my students, they say they're getting some really interesting results. And then a parking lot conversation that, and I kid you not, you're like, and that's something that you must do, is chat to people wherever. So you all know Brian Moritz from BCB. We had a chat about reptiles, and suddenly I said, what do they know about reptile muscle? And he said, nothing. And I did some research, nothing. And here's something that I didn't know. Reptiles don't stop growing. I didn't know that. Some do, but some don't. So we're going to start looking at reptile skeletal muscle, because if we can find out what keeps them growing, we could potentially extrapolate that into human research, or to extrapolate to humans. The little lamb there is only for show. So I'm going to wrap up now, and I think those people, those 6 people, Kathy Meyberg, Bengt Saltien, Jesper Anderson, Birgitta Essen-Gustafsson, Edward Ojuka, Prof. Tim Noakes, have been amazing. And it's so phenomenal to have those people part of your life and just to be human with one another. And we're all human, we all share the same pain, we share the same goals and dreams. And thank you for that. Ed, if you're listening, thanks, man. And Brigitte, thank you so much. And Jesper as well. Kathy, of course. Bank Saltine sadly passed away just before he sent me a booklet on camel muscle. He sadly passed away a month after that. To all the MyoLab peeps, I want to say thank you so much for bringing joy to our laboratory. You are really phenomenal. These are over the years. I really appreciate you, and thank you for making my journey so pleasant. To the current peeps, and we're going to grow, thank you very much, and keep up the good work. And don't think small, think big. And of course, all the collaborators and friends over the years, thank you so much. You've really made science exciting, and it's so wonderful sitting around a campfire in Nelspruit, and we can have a beer or a whiskey or a cool drink, whatever, if you're pregnant, and then chat about what is the future going to hold. And then I think I just need to mention my family that I've lost over the years. My mother, she was a quite tannie, but she's really pushed hard to bring the best out of me. You saw that gumboots kid. He was a very naughty child. My dad, who was this soft-spoken and very wise person, super smart, and my sister that sadly also passed away. So I give them credit and honor them in this road. And then my current family. And there my mother-in-law just jumped up and down, yes. And I want to thank you, my Joe, Linda, for your amazing time and just being such a great parents-in-law. If you get married, that's what you want, great parents-in-law. My wife, I love you to bits. And my kids, hopefully you're not embarrassed now. You're at that age where you're embarrassed by your dad. And yeah. And with that, I'd like to say thank you. And all the funding and support that we got over the years to pursue research that has been such an amazing, phenomenal journey. Thank you very much.
Please, thank you for that excellent talk, Professor Kohn. I will call you again. I see you were waiting for something, but I will call you again soon. So, I now call upon Professor Tim Noakes to respond to this inaugural address. Professor Noakes, you're welcome. Thank you.
Deputy Vice-Chancellor, Deputy Deans, everyone else, thank you so much for inviting me to do this great honor. Thank you. No, I'm going to cry. What you witnessed is one of the best lectures I've ever heard from a new professor. It's absolutely stunning. And it's unfair for me to now have to come and talk after that. So it was just amazing. We're so proud of you, Tertius. And thank you to the University of Western Cape for making a home for you. And you know what that means, and maybe I'll discuss that. Why was it so good? I mean, the quality of the quality of the slides, the content, and the way it was presented was absolutely flawless. It couldn't have been better. And I think anyone who watched it would be able to follow it. And this was complex stuff that you were talking about. Point one, no one else could have given that lecture anywhere, and no one else in the world could have given that lecture. And I mean that. And that's why we're so proud of you and of the University of Western Cape for recognizing you as what you are. So I'm absolutely speechless. It was so good. So I'm going to just so I can't add anything to that lecture. I couldn't have given it. I could not have ever given that lecture as well as you did. And it was lovely to see some of those people, because what you don't know was that in 1975, that when you were born, I just finished my internship at the university at Groote Hospital, and I realized I was never going to be a doctor, that I couldn't be a practicing doctor. It just wasn't in me. And I decided I had to go into science. And I went to a conference in New York in 1976, and guess who the first speaker was? It was Birgitte Essen-Gustafsson and Bengt Saltine. And I was so overwhelmed by what they said, I said, that's what I'm going to do. But I didn't have the skills that you have. And so I couldn't do it. I had to do other things. And I'll explain that in due course. So I've known Tertius for at least 20 years when he was a postdoc at UCT. And more recently, when I met him, you know, he was just getting into the area, and which I was really interested in. You mentioned that I kind of helped you. And then you ran it to this, where was the money going to come from? So I'm not trying to say anything particularly important about myself or my wife, but this is the fact. The fact is I wrote a book called Law of Running, and it had accumulated money over 20 or 30 years, which we put into a fund. And I wanted to use that money to fund a really good scientist. And Tertius was the first person we funded. And so, so, I mean, I had absolutely no idea how good Tertius would become. I mean, how could I know that one day he would be a full professor at the University of Western Cape? So why did we choose to support him? That's the question. And I think this is what I want to talk about. He's given you the science, but there's a person behind the science, and that's what professors must be as well. They must be exemplary, absolutely exemplary. And there are a few people I think are more exemplary than he is. And so I'd like to, for his family as well and his children, just make you understand how fortunate you are to have this man as your father. So, what were the values? So I realized that he was a good person, and good people do good research and become productive people in society. And what were the values that he espoused and which I could pick up early? Firstly, he's absolutely honest. Well, he tells you that's what it is. You don't have to worry that there's anything else that could be untrue about him. So what he says is the truth. He's absolutely transparent. He hides nothing. And that is so uncommon today in today's world. And he doesn't have to be not transparent because he's so good at what he does. The people who are not transparent aren't good in what they do. There was the transparency, the love of his family, that was obvious, that came right through in his talk. And that was, he'd always expressed that when I was with him. He has a very strong religious faith, and I think that that is so important today, in today's world. It's terribly, terribly important, and we're losing it, unfortunately, or it's coming back again, perhaps, because of what we've been through. Then what he didn't talk to you about is his courage. And the reason he's at UWC and not at UCT was because he was targeted in the most vicious, horrible way. And it was, for both of us, it was a terrible time. And he chose to fight, and that's why he could come here with his equipment and continue his life. And again, I thank UWC for taking him, for accepting him. Because what happened to him was totally, totally unacceptable. And it would have destroyed other people, but he stood firm. To be a great scientist, you have to be inquisitive and curious, and I think that was one of the keystones that he spoke about today, is how curious he is. I mean, the range of the work that he did, I wasn't aware of all the publications that he has. I mean, I follow very closely what he does, but I was absolutely astonished at a number of publications he has, and the quality and the extent. I mean, there's no one else who has that range in the field, who has that range of publications. No one in the world. So that's what you're dealing with. And it's so fantastic to have someone in South Africa who is a leader in the world. His ranking, by the way, in the NRF is a joke. He's not a C2. So it's absolutely ridiculous. He's a high B. He's a high B. Yeah, and he will become an A-rated scientist in due course. Yeah. And the other thing that you might not have picked up is his technical abilities. Those slides, producing the stains and the work that he did, it's phenomenal. I was working with a friend of mine who's a surgeon, and I was asked to write something about him. And a friend of his said that only 10% of surgeons are uniquely skilled. Uniquely skilled. The others are sort of, they get by. But there's a group who are uniquely skilled. And they save all of us. Those are the ones you need to get hold of. And he is uniquely skilled in what he does. So for all the other great advantages he has, his laboratory skills are phenomenal. When he spoke about those slides where he measured the LDH or whatever CS in the different fibers, I don't know how you do that. I mean, the samples are so tiny. To be able to measure the enzyme activities in those fibers it's just remarkable. And you need to come and watch him do the contractility studies of these tiny fibers. I mean, how he even sets the system up. You know, there can't be there are only a couple of people, probably 3 or 4 in the world who do it. 3 or 4 who do it, and he's one of them. Yeah. So I don't think that he made, because he doesn't sell himself, he doesn't have to, because he's done the work. People don't understand the extent of his genius and what he does. And I hope that I can get that over. He's focused and totally committed, that's very clear. And then he spoke about his leadership, and I want to watch some of those Nelsprad experiments where they chased the Blesbok around. And by the way, the Blesbok are very good, and they're very strong, and they're very resilient, so they can do that running. It looks like it's kind of harming them, but it's not. They're built for that. And so I saw that, and that was phenomenal, his leadership in setting that up. You saw how many people around the world came to do that work. So I don't want to take any more of your time. I just want to say that it's been a huge privilege tonight. It's unbelievable. As I've said, it was one of the great moments in my career to hear you speak tonight. And your career's only just started. What we saw today is just the beginning. And I hope I live a little longer so I can see where you get to, because it's going to be very, very exciting. And again, thank you to all the senior members of the university for giving this man the opportunity to be not just great, but to be very, very special. He's a very special man, and he needs everyone to support him. And he never asked for it, he just gets out and does it. So congratulations, Tertius, and to your family. Wishing you all many years of happiness ahead. And we're just so proud to be associated. Thanks very much.
Thank you, Professor Tim Noakes, for that wonderful response. I think this is probably the moment you are waiting for, Professor Kohn. I'll just call upon our DVC as well as the Dean just to hand you something to acknowledge your address. And Professor Tim Noakes. And Professor Tim Noakes, if you may also come down. We also have something to acknowledge your response. Thank you. DVC as well. Thank you. Okay, then we're almost coming to the end. It's time for a vote of thanks, and I invite our Deputy Dean for Research in the Faculty of Natural Sciences, Professor Fanelwa Ajayi, to give a vote of thanks. Thank you.
Good evening, ladies and gentlemen. As Professor Kohn was speaking now, I was just thinking, where was he when I got 0 out of 20 for my first tutorial? Because I came into medical biosciences. So that's when I knew that I have no future here. My very first tutorial at UWC at the MBS department was a 0. And I went to the lecturer to ask if they didn't make a mistake. And then he brought out the memo to show me exactly that you got everything wrong. And he didn't understand how I got everything wrong. So I thought, you know what? I have no future. So I only hope and I only wish that you were there, because I may have stayed and following your steps. So I just want to thank you very much for today. It is an honor to have you in the faculty. And the type of scholarship that you contribute to the faculty is great. And I want to thank you very much for today. I want to thank you also for your contribution. You may not know because you do the work, but when you submitted your professorship documents, your portfolio, wow, I just thought, how does this man have a family, have a life outside of this UWC if you can be able to deliver such work? So congratulations, and thank you very much for today. Thank you very much. Colleagues, we have a rector who has a good vision for us, and I want to thank him this evening because us being here today and to celebrate Professor Kohn is also a vision of his and a vision that I think UWC is moving towards, that we need to celebrate our scholars, we need to celebrate excellence. So thanks to him. DVC, Professor Matiba, thank you very much for being here today and for supporting us as a faculty in this endeavor. Thank you very much. I also want to thank our program director today, Professor Maya Dube, for carrying us through. I realized that he was supposed to sit where I was sitting, so I'm sorry, my friend, but you have to run around. Today was your day to run around, so thank you. Ms. Kelly Williams, thank you very much. I don't know where she's sitting. She sang very well, and thank you very much for the anthem rendition this evening. And also to the institutional advancement, the banners that we have here advertising today's event, as well as the printing of the programs. We want to thank them as well for making today extra special. Ms. Tasneem Parker, she works with the Dean. She's the Dean's Secretary, and she's the one that does the running around to make sure that the event today went well. So thank you very much. I don't know where she is. She's probably outside. So thank you to her. The members of the academic procession as well, thank you very much for being here this evening and to support us as well. Colleagues, thank you very much. But I think I want to say thank you to Tertius family, to the children, because, having a busy dad and to the wife, to have a busy husband is not easy. But the support that you give him, we are appreciative because his contribution is because he's grounded, both in his family life as well as in his friendships. People that are in his life, his students, it means that there's support. And I appeal today, it's not easy being an academic, and I appeal that let's hold hands and let's make sure that we support each other. It's not easy to be alone and walk in this academic journey. So thank you to the family and friends. And the guests, thank you for making tonight very special. It's very nice, to stand and to see an audience when you speak. So thank you very much for being here. And please continue to support these events as they carry on in the faculty. So thank you very much. Thank you very much for your time and your support. So I give over to you, Program Director, again, for the last time, please come forward. Thank you very much, everyone.
Thank you, Prof. Fanelwa, for that vote of thanks. So yes, this brings us to the end, and it's now a time for refreshments and photographs upstairs on the foyer. So we can all proceed to the foyer, so we may all stand and let the academic procession go ahead first, and then we can all follow to go upstairs for refreshments. Thank you very much.